Well, I am really thrilled for this new Ground Truth live video to welcome a pioneering neurosurgeon who's the CEO of the Feinstein Institute of Medical Research at Northwell, Dr. Kevin Tracy. Kevin, it's great to have you.
It's great to be here, Eric. Thank you for having me on. I'm looking forward to this chat with you.
Yeah, well, your work, which has been going on for decades, culminated in this book. A part of it encapsulated in this book, The Great Nerve. If everybody doesn't know what the great nerve is, it's the vagus nerve. Vagus for Latin word for wandering, and this nerve is like the internet throughout the whole body, connecting the brain to everything. And you made a seminal discovery. I think it was back in 1986 with baboons. The idea that up till that time, the brain and the immune system were thought to be in different orbits. They weren't connected. They were firewalled. And you came up with experiments, I think, in baboons and tumor necrosis factor. And for the first time, you connected the dots. Is that a fair summary of what happened back then?
It is. It is a fair summary of what happened. Back in the 80s, I was part of the team here in a few blocks from where I'm sitting in New York City at Rockefeller and Cornell that identified the biological activity of TNF as a major mediator of he first demonstrated that monoclonal antibodies targeting TNF could inhibit inflammation. Now, this was published back in Nature in 1987 in a paper I wrote with my colleagues. And this paper was the first description of monoclonal anti-TNF antibodies as having a therapeutic potential. And of course, today those antibodies have all kinds of commercial names like Humira and Remicade, and they're related ones for IL-1 and we call the whole thing cytokine storm. But back when we were doing this work, what we were actually defining was that TNF is sort of the proximal trigger of a cytokine storm. And today that's treated with these monoclonal antibodies. But the real question that remained is if the body has the ability to produce these molecules, cytokines, TNF, other things, that can cause so much damage through inflammation, then how is it normally controlled? What is it that evolution did over millions of years to put the brakes on inflammation and prevent it from causing damage in most of us who are healthy? And that question led to the discovery of the role of the vagus nerve acting like the brakes on your car to suppress inflammation.
Yeah, so I think this was such a big thing because this inflammatory reflex, as you labeled, I think, really brought together the pathway in the brain, this great nerve, vagus nerve, as a mediator of inflammation throughout the body, which sets up a whole lot of ability to hack into this vagus nerve with vagus nerve stimulation. And so you present in the book many anecdotes. Before I get into that, when you made that discovery back in 86, a lot of people call it a eureka moment. You called it a holy shit moment.
So the holy shit moment came in the 1990s.
Oh,
We were specifically addressing the question, how might the brain, by sending signals through the vagus nerve, be turning off inflammation? And that came about because of an experiment that produced a big surprise, and you could call it an accident in the lab. We were putting an anti-inflammatory molecule into the brains of mice and rats with the sole purpose of blocking inflammation in the brain in order to reduce the damage of a stroke, an experimental infarct. And that all went as planned. What we hadn't planned is that when we put the anti-inflammatory molecule in the brain, and we had developed this molecule in the lab, so it was an experimental drug. When we put this in the brains of these animals, it also turned off inflammation in the body. That was the holy shit moment. Like, how could the brain be sending signals to control the immune system? There was no way in 1998 to explain this. We went into the literature, and I found an old paper, then an old paper, now even older, that had been cited by essentially nobody. That Linda Watkins from Boulder, Colorado, had been studying sickness behavior, and she showed that when she gave IL-1, a cytokine, of course, into the abdomen of animals, they develop sickness behavior, anorexia, fatigue, behavioral withdrawal, social withdrawal. And when she cut the vagus nerve of those animals, it went away.
mhm.
And so the only way to explain that was that the vagus nerve was sending signals up into the brain that was activating the sickness behavior networks. I got to thinking maybe when we had put this molecule in the brain of our animals, maybe we had discovered the motor arc of a reflex, and Linda had discovered the sensory arc of the reflex. And so I wrote a hypothesis paper, published it in Nature, called The Inflammatory Reflex, and it turned out to be correct. After 20 years of mechanistic work, we've connected all those dots.
Yeah, no, it's amazing. We're going to get into the rheumatoid arthritis, both the trial that you did, remarkable trial with a sham control, and the approval by the FDA, which it really tells us that the progress is finally, you passed this threshold, and decades of work is really together. But before we do that, you go through some remarkable patient anecdotes in the Great Nerve book. One that is so striking is Kelly Owens. Can you tell us about her story?
Kelly is a remarkable human being who, as a teenager, was diagnosed with inflammatory bowel disease, Crohn's disease, which was devastating for her. Um, Not only did it put her in and out of the hospital for many, many years, it also forced her to be subjected to use suppressive medications. And a whole list of biologic agents like anti-TNF we were just talking about, all of which have in common, many of which have in common, they have black box warnings, which are the most serious side effects that FDA will label of a drug. I mean, the side effects include sepsis, cancer, tuberculosis, fungal infection, and they're invasive. These biologic drugs are invasive. And in Kelly's case, they didn't help her. Some people say she failed the drugs. As a physician, I never say that. I say the drugs failed the patient.
Right, right.
And her symptoms, in addition to having GI symptoms, for many years. They also, as can happen in some Crohn's patients, it also gave her a severe form of arthritis and arthropathy, which intermittently required her to be in and out of wheelchairs and in and out of a cane. And this went on for many, many years. And finally, her physicians told her to get used to a life of steroids. And around that point of time, she saw me on Huffington Post Live, and I was talking about this vagus nerve story that you've introduced, and I was talking about the fact that we were launching through a company that I co-founded. The company was launching a clinical trial in Europe, in Amsterdam, and in Bosnia and Croatia. Well, Kelly and her husband Sean talked their way into that trial somehow. They sold everything, as Kelly says, everything that wasn't tied down in New Jersey, they sold. They did a GoFundMe sort of campaign with friends and family, and they moved to Amsterdam for 6 months. She was implanted with what I call a Gen 1 vagus nerve stimulator, and we can talk about Gen 1 and Gen 2. And when it was turned on, within 2 weeks, she felt a lot better. And a couple weeks after that, she was on her way to a follow up appointment in Amsterdam and saw the elevated truck. He gets downstairs and is looking around like, where the heck is Sean? And Sean is standing at the bottom of the stairs with tears of joy running down his cheeks because he hadn't seen Kelly run in years, especially not run up a flight of stairs. He was used to her not even be able to walk up a flight of stairs. So Kelly, that was about 8 years ago. I've been in touch with Kelly many times since. She worked with us for a while, collecting a database of patients who reach out to us interested in this therapy or in clinical trials. And she takes no medications. Her only therapy is the vagus nerve stimulator, which is activated for 2 or 3 um, times a day for 5 minutes. And she asked me once, "Am I cured?" And I said, "Well, Kelly, that's a good question, but you're asking the wrong guy. Other than my conflict of interest, I'm not your physician." But it
Right.
really raises the question if readjusting her vagus nerve activity enabled her nervous system through neuroplasticity to reset the set point on her immune response. And it's an open question. But yes, she is symptomatically relieved and takes no medications and is doing great.
Yeah, well, she attributes you to saving her life, and it's pretty impressive. Now, there's another story. I mean, of so many, but the first person, Pedro or Piero Droge, who was with rheumatoid arthritis, who was treated with vagal nerve stimulation. Tell us a bit about him.
So Pero lives in um, Mostar, Bosnia, in the Dinaric Alps in the west of Bosnia. And that was a trial site. And as it turned out, as you know, doing clinical trials, you set up a multi-center study and someone's first somewhere, and it happened to be Pero. So it was back in 20, and I got word through Setpoint that Setpoint had launched the trial and flown a neurosurgeon over to do the surgery in Mostar, and that the patient was will. And I said, "Can I go meet the patient?" And they said the patient would love to meet you. So it was Thanksgiving week of 2011. I flew over there and learned that the patient had received his Gen 1 vagus nerve stimulator on Labor Day of that year, so the first Monday of September. And he had spent, he told me, years homebound on his couch. He was a young guy at the time, and had young kids, but he couldn't play with his children, couldn't drive his truck, couldn't work. And his doctors at that time in Bosnia, and so he had not responded to steroids and methotrexate. Well, he had a very significant clinical benefit from his implant, vagus nerve stimulator implant, and was out playing ping pong a couple weeks later. I didn't know until then, but ping pong is huge in Bosnia apparently, and he felt so much better playing ping pong. He went out and started playing tennis. Promptly injured his knee because he was completely deconditioned from being on the couch for years. So now, because the swollen knee impacts your clinical assessment in an arthritis trial, his doctor said, take it easy, no more sports until the trial is over. So I called in the course of writing this book, The Great Nerve. I called his doctors and I spoke to them about a year ago, and he also is doing great. He's taking no medications and has no symptoms. He's driving a truck. He's unloading cargo, and we would say he's in remission.
You know, it's fantastic. Now, of course, these are anecdotes. They're really striking, but the real test, of course, the acid test is to do the randomized trial. You did that. It was published in Nature Medicine in December. We'll put in the links when this is archived. But that trial was impressive because it was done very rigorous with a sham control, hundreds of people in each arm, and this was in rheumatoid arthritis. So prior to the publication, even the FDA gave approval this just last year, months ago, for the first time for a inflammatory autoimmune disease, that vagus nerve stimulation was given FDA approval. So, can you take us through? This was a big deal. This was a culmination of many decades of effort on your part. Yeah,
Yes, you're absolutely right. My part and the part of hundreds of people in laboratories, my laboratory and other labs around the world, but also hundreds of people who work directly or indirectly with SetPoint, who developed the device for the trial and raised all the money to pay for everything and did all the regulatory work. So um, it's really important to point out that there's been two clinical trials. The first one was published in the Proceedings of the National Academy of Sciences around 2016. I was a co-author on that, and that was a smaller open label trial that was led by Paul Peter Tack and his colleagues at Amsterdam Medical Center under the support and sponsorship of SetPoint Medical, and the chief medical officer then was Dr. Ralph Zitnik. Now, my colleague Sangeeta Shivan and I studied the first patients that were implanted at our hospital here at Northwell Health in New York. And Ash Mehta, the neurosurgeon, implanted these patients. And the first question in that first trial, 2016, was if we turn on the vagus nerve stimulator, can we activate the inflammatory reflex? And the answer was yes. When we implanted the device, measured collected blood before the implant and after the implant, but before it was turned on, and then collected blood again after the implant was turned on, there was a statistically significant reduction in the TNF ability of the white blood cells of those patients to make TNF in response to endotoxin. So that was really important. And then the open label trial had a positive signal in rheumatoid arthritis. That laid the groundwork for the FDA to grant SetPoint a breakthrough designation, which was turns out now to be very important, as you know, in these regulated times, and laid the groundwork for the labeling trial. If you're in the drug business, you would call it a phase 3 equivalent trial. This is a device, so it's the definitive trial that was set up as you describe. That trial, I was not a co-author on that trial. That was an independent trial done at 40 sites across the United States. It was led by John Tesser from Arizona and by Dave Chernoff from the chief medical officer at SetPoint. The study enrolled, as you said, 242 patients, and all of them had refractory rheumatoid arthritis, which means they were having serious signs and symptoms despite the fact that they tried these powerful immunosuppressive drugs we talked about, the biologics or the JAK inhibitors. So these people were sort of out of options,
yeah,
and they enrolled in the trial. Now, there's a really important point about this. Um, 242 patients made it into the trial. Guess how many created a portal, logged in, and put their information in, trying to get into the trial?
probably thousands, right?
30,000.
30,000. Wow.
Thir 30,000 people tried to enroll in this trial for 250 spots that ended up with 242 patients in the final readout. And the reason is the and this is often overlooked when people talk about these kinds of things. The drugs are invasive.
Yeah.
You're, they're all the biologics are injected.
City. I mean, my goodness, yeah. And, these are very expensive, have, just potentially very serious side effects. I mean, you've already run through a couple of the people that were demonstrative of that. So yeah, I didn't know 30,000. That's amazing. But what is the surgery involved to put in the device?
The device, I'll pull one out here. The surgery involves about an inch and a half incision in the left neck at about the level of the Adam's apple, the larynx, to implant a device that's about the size of a multivitamin or a fish oil pill. The device sits directly on the vagus nerve at about the level of the carotid pulse. You can feel your pulse. And it is self-contained with those two shiny things are the leads that sit on the vagus nerve. There is a computer disc, of course. There's an antenna to talk to the doctor's tablet. I always drop it when I'm at the podium, and that's very complicated on stage, Eric. It also has, which is really clever, It sits on the vagus nerve, but it's wrapped in this silastic, like a pea pod. And so the nerve runs underneath it, and then there's a stitch that goes through the top to hold the whole thing in place.
How long
And
did that surgery take to put it in?
an experienced neurosurgeon will put it in under an hour, anywhere between a half hour and an hour. It's an in and out surgery, outpatient. Come in the morning, go home in the afternoon. We've been doing them in the hospital, but obviously this will be amenable in the future to a full outpatient standalone facility. Right now, neurosurgeons are being trained by the company in the procedure, and so there's a select group of neurosurgeons who are doing this at select sites. But eventually, again, it's possible. I don't know this. I'm not an insider in SetPoint Medical, the company, but eventually, I would imagine a time when the implants are brought into other specialties once the expertise has been gained.
Sure. Now that device gets stimulated a couple of times a day.
So the device has a rechargeable battery. So the doctor to start, because the FDA has a this for rheumatoid arthritis, the rheumatologist will program the device to turn itself on for 1 minute a day. Nothing for the patient to do. When the current is flowing, I've
They
met
feel
many
nothing.
many patients.
They don't feel anything when it's stimulating.
Some patients feel a little tingling for 1 minute in their neck. Some patients feel nothing. I've met people who sleep through the therapy once a day, and I've met other people. I met a school teacher, and she was written about in the New York Times, Dawn, and she told me that it wakes her up in the morning. And I said, "Oh, that's too bad, 4:30, waking you up at 4:30." She goes, "No, I get up early anyways. I don't mind it." And for years, my alarm, I'm a teacher, and my alarm would go off at 4:30, and I would feel the pain in my hands and wrists, and I'd start to cry. And now this thing wakes me up with a little tingling, and I wake up and I have no pain in my hands, and I get a big smile on my face to start the day. So she doesn't mind the tingling.
Well, that was good because you had a sham control where you put in the device and half the
Abs-
and
That's
of course, it wasn't getting any stimulation. It was just the device was in. That's the ultimate control in a device randomized trial, right?
That's right. That's exactly right. And as you know, the data are all in the Nature Medicine paper that John Tesser is the first author. And what that was an important factor in the sham controls. How many people in the sham group think they have the active group? Think that they're feeling it come on for a minute a day, and that it was a small percentage. That was almost the exact same percentage of people in the active group who thought they could feel it.
Yeah, yeah, yeah.
So it's pretty. There's no perfect control for a procedure like this, obviously, but that's pretty darn close to perfect.
Oh, that's impressive. Now, one of the people was asking, "What's the name of the device?" It's a VNS, a vagal nerve stimulator. Is there a name to this?
The set point device is called an immunoregulator,
Okay.
technically it goes on the vagus nerve and puts electric pulses into the vagus nerve. So yes, it's a vagus nerve stimulator, but it's very different from what's been used for 40 years to treat epilepsy and depression. So the devices are different and the signals are different, but the biggest difference we could talk about first is that the signals that are used to treat epilepsy and depression by historic precedent, those signals are on for 5 minutes at a time, then off for 5 minutes, 24 hours a day, 365 days a year. So there's a 12-hour duty cycle at 5 milliamps, as opposed to a 1 minute duty cycle in 24 hours at 3 to 400 microamps.
Oh, wow.
And the reason is we understand the biology of how the inflammatory reflex downregulates cytokine storm. And the 1 minute activation in the neck drives signals down into the ganglia of the abdomen, which are relayed into the splenic nerve and the spleen. And at every step in the sequence, from the vagus nerve to the ganglia, from the ganglia to the splenic nerve, from the splenic nerve to the T cells, and from the T cells to the monocytes, you have this thousandfold amplification of the signal, and you have a persistence of the signal that goes on for hours and hours afterwards with neurotransmitter release that suppresses the cytokine. So 1 minute gives you 12 hours of protection.
Amazing, really. 1 minute a day. Now, why don't we have for all autoimmune diseases? Why isn't this become a frontline therapy instead of all the drugs?
Well, you're
Yeah.
not going to get me to say all for anything in medicine, right? Nothing's going to work for everybody all the
Sure.
time, but you're going to
Op-
see, yeah.
Option.
I think you're going to see a lot more clinical trials. So we've seen very impressive open label trials for inflammatory bowel disease. It works. We've seen very impressive open label trials in some diabetes models and type 2 diabetes studies. And we've seen very impressive preclinical results in, again, diabetes, in multiple sclerosis. We've seen pilot clinical trials in lupus arthritis. We've seen pilot clinical trials in other inflammatory conditions. So I am agreeing with you 100%. This is the tip of the iceberg. There's work to do. There's clinical trials to be done. There's going to be money to be raised and spent. But I think you're going to see it happen because patients are going to demand this. I mean, we talked about the expense of biologics. We talked about their invasiveness. We talked about their immunosuppressive black box warnings. We left out the fact they only work half the time.
Oh,
So
that much actually.
yeah,
That
40%, 50% if you're
yeah,
lucky, right?
yeah.
So the demand is there, and I wrote this book for the simple reason of establishing a platform of basic information for patients, for doctors, and for family and friends interested in the vagus nerve. That this is what we know to be true in the lab and in the clinic. This is what we know to be false. And these are the important things that I'm urging my colleagues to double down and work harder on because we've got a lot more to do. I think we're going to help millions of people.
Well, I think the autoimmune diseases that you just touched on, I mean, they all deserve study. The rheumatoid arthritis results are so striking that in refractory people, no less potentially to avoid immunosuppression and all these other side effects, it has a lot of potential. Now, one of the questions that one of the listeners brought up was about an external device, and that brings me to long COVID. So POTS, as you know very well, postural orthostatic tachycardia syndrome, very prominent among people with long COVID. Along the way, there was a company in Canada that was an external vagal nerve stimulator. Has this been tested? That appears to be inflammation directly of this nervous system. What has there been any experience with external devices and or this device in POTS?
Um, there's been experience, but to my review of the literature, and I'm speaking to the world's expert on the review of the medical literature, so that makes myself nervous. But that being said, my review of the literature, I am unable to find for handheld devices applied to the skin any evidence that any of them directly stimulate the vagus nerve. So as biomedical engineers, we've tried to do it in my lab, and we tried to do it in volunteers and biomedic. We've worked with biomedical engineers. If you put electricity on the skin of a human being or a large animal. The vagus nerve in humans in the neck, where you pointed, is deep in the neck. It's wrapped in its own sheath with the carotid artery. It's underneath several layers of fascia. It's underneath the platysma muscle. It's underneath the skin. Electric current doesn't travel like a laser beam where you point the thing when you put it on your skin. It doesn't work that way. And so it's okay to call things um, vagus nerve stimulators um, after they've gone through FDA trials, but it doesn't mean they are FDA,
Yeah,
that they are vagus nerve stimulators.
you're just not going to get it right from an external device. Is I think what you're saying, and this is something has to be right on the vagus, right?
There's one place the vagus nerve sends a branch to the skin, and that is in the cartilage of the external ear called the symbiconcha, which is the part of your ear that looks like a seashell around the open ear, symbiconcha. Now, if you electrically stimulate the symbiconcha with some sort of device, a TENS unit or something, then arguably you've stimulated the vagus nerve because you've stimulated the sensory fibers traveling from that cartilage. You know why it's there, right? This vagus, it's a vestigial branch of the vagus nerve that used to innervate evolutionarily the cartilaginous gills of fish. And so it dragged the nerve with it when that cartilage became the cartilage of the external ear. So are you stimulating the vagus nerve when you put electricity on your yes, and there's some really interesting, and I'm a fan of it, right? There's some really interesting science that's been done with brain imaging, fMRIs, far field evoke responses, and you can connect all kinds of dots, but you can't say it's the same thing that SetPoint is doing as putting a device on the vagus nerve to specifically activate vagus nerve fibers. So what does that mean? It means if you look at the typical study of an externally used device, whether it's on the neck or the ear, what you see are relatively small numbers of subjects, usually 20 or 50, usually open label,
Right.
usually not well controlled, and usually measuring some clinical effect, which often reaches statistical significance, decreased inflammation, increased heart rate variability. This is interesting stuff, Eric, but it doesn't prove the mechanism.
Yeah.
And it doesn't prove the mechanism, and almost none of it, even some of the trials that have led to FDA approvals for various devices, almost none have been replicated in a way that would make you comfortable to recommend these things for your practice or would make your professional societies comfortable recommending it as a broad scale thing, meaning they have not been subjected to large, randomized, well controlled, statistically rigorous clinical
Right.
trials. That's what
Well, maybe.
I'm not opposed to these things.
Yeah.
I I do it. I use a TENS unit in my ear most days because it might work, but I don't recommend it to people as a therapy because I'm not sure it does work. How's that?
Yeah, no, I get that, and, that you're bringing up the other parts of the story here with the TENS device that a lot of people are using for serious pain, and, also the fact that this got its start, not related to relieving inflammation, but it was back in epilepsy, 1988. VNS was used for epilepsy, and before we got started, you were telling me about how, and it's in the book, how people it helped some people with epilepsy, with their seizure control, but the people didn't wanna have the device taken out who it wasn't helping. Why was
Yeah.
that?
Yeah, so there's 2 or 3 interesting stories here. Like you, I'm a fan of medical history, but the story begins at the end of the 19th century when a New York physician started treating his patients with epilepsy, serious seizure disorders, started treating them with like a dog collar-like device that he would put around the neck of his patients, connected to a Faraday charging coil. And so he was shocking these people. They had to be tied down on his exam table. So that's where this all started. And there's some great treatises of those stories that are out there. And so skip fast forward to the 1980s, as you said, and well, actually, right after World War II, in Pisa, Italy, a physiologist worked on an epilepsy model in his lab. I believe it was in cats, and showed that if he induced seizure activity in the brain of the cats and measured it by EEG, but applied electric current, just brief electric pulses to the vagus nerve of those cats, that it would stop the epileptic spikes. So in the 1980s, a Philadelphia neuroscientist patented that, cited that Italian's work in his patent, and led to the first clinical trials by a company called Cyberonics. The first device was implanted by a neurosurgeon named Bill Bell, who was actually my chief resident when I was an intern here at the New York Hospital in Neurosurgery. And it turned out to have very significant benefit, um, helping some patients who would go from having 10 or 20 or 50 seizures an hour to having none or very few. I mean, putting some people who were homebound back at work and out. And today we don't know why. I pretty much just told you most of the science that we know about vagus nerve stimulation and epilepsy. It seems to work in half the people, but we don't know why. The other half that it didn't work in this back in the early days, the surgeons would say, "Well, this isn't helping you." Well, take it out. Patients like, "You're not taking out my device." Surprising the doctors, and they asked why, and the patient says, "Because I feel better. It makes me happy. It makes me." Based on that, that led to clinical trials of using vagus nerve stimulation to treat treatment refractory depression patients, patients who are out of options with talk therapy, patients who are out of options with medical therapy. And as it turns out, in years and years of work, it works again about 50% of the time, about half. And people say, well, I wish it was more than 50%. Well, some of the people that have benefited from these vagus nerve implants for treatment resistant depression have gone from being suicidal or unable to care for their families or unable to work to being back in the mainstream. And so I wish there was a lot more talk about this, Eric. I wish a lot more people knew about this. I wish we knew a lot more about the mechanism, and I wish we had really better analysis of the cost benefit ratio, because I think more people should know about this and more should be done.
Yeah, I think it's important to point out there the randomized trial in refractory depression was just published this month. And it's, as you say, half the people derive substantial benefit with severe refractory depression. So, this whole idea of hacking the vagus nerve, whether it's to reduce inflammation or maybe not so clear cut as far as the mechanism for benefit, it's pretty striking the diverse beneficial potential use cases, if you will, clinical applications. Now, one of the things that is hot these days, which I don't understand, are cold plunges. You have these people advocating, like, Peter Attia and others of the bro science. You should go for cold plunges. And these people are going from cold plunges to hot saunas. There's a diving reflex. What's going on here? Is there anything to cold plunges?
We could talk about this for 2 hours, Eric. I'm not sure we could do that. I think you have to be really careful when you talk about what we understand about these physiological responses because it's complicated. So maybe we could back up for 1 second to the vagus nerve. Many of your listeners are physicians, and many of your listeners are scientists and well versed in all this. But for those that are not, we say vagus nerve, but you have two of them. You have one on each side of your neck, and we say two vagus nerves, but inside of each of them you have 100,000 fibers. So you have 200,000 vagus nerves technically, and each and every one of those individual fibers was selected for and subjected to evolutionary pressure for millions and millions of years to have a specific origin, a specific destination from the brain to the body or the body to the brain, and to do a specific function. And the idea when we stimulate in the lab and with our limited clinical evidence, the evidence suggests that when the idea is to control inflammation, we're stimulating a few hundred fibers, and that's sufficient to control inflammation. Those are not the same fibers that control your response to the cold. Those are not the same fibers that control the response to your heart. Those are not the same fibers that control your response to breathing. So the question is, when someone tells me I want to do this, that, or the other thing to stimulate my vagus nerve, I say, oh, really? Which one are you going to stimulate? And how are you going to do that? That's where it gets complicated. It's incredibly complicated. So, cold plunge, everyone remembers their first cold plunge, right? And because it's awful. I mean, it might happen by accident. My brother tricked me on a warm spring day. My brother tricked me into running into the freezing cold ocean. He was standing at his ankles and he told me to do a running dive. And when you go into that 50-degree water, um, you come up and you can't breathe. All the air goes out of your lungs and you can't inhale. And that is a massive fight or flight response. That is a massive fight or flight response. You have a surge of epinephrine and norepinephrine. Your heart rate goes up, your blood pressure goes up. And that's the opposite, allegedly, of a parasympathetic fight or rest and digest response. But what happens if you stay in the cold water and you cold adapt? And here, Eric, it's very hard to find well controlled um,
Oh, really? Yeah.
clinical studies. I found a few, and they're in the book, but it's very hard. Usually, it's military types, soldiers in Iceland or something, or in the Baltic states, getting sprayed with water in a cold room and with dressed in a bathing suit, and then they give their blood. And so there's some data like this. If you cold adapt to some point for some period of time, you will actually feel your heart rate slow down. At which point you can say, my vagus response is exceeding my fight or flight response. They're both still active, by the
Yeah.
way. But
Yeah.
but let's just say for that you've tipped from primarily sympathetic to primarily parasympathetic. So okay, now you've stimulated your vagus nerve, but you've done a million other things to your nervous system
Yeah.
too. You haven't put a device on your neck targeting a few hundred fibers to turn off inflammation. Now, do the fibers that made your blood pressure go up first and then down, and your heart rate go up first and then go down? Are those the same fibers that go to your spleen and control inflammation? I don't think so. Does the whole thing act as a on block everything at once response? I don't think so. I can play the piano and chew gum at the same time. So when you break it down in a sequential way, you can understand why people are interested in it. And I do cold showers several times a week at the end of my regular shower. And the reason is fight or flight, acute controlled fight or flight is anti-inflammatory. We and many others have proved that, including Wim Hof, by the way. And increasing vagus activity is anti-inflammatory, and we proved that. So for me, it's a Pascal's wager, right? My doctor says I'm okay in cold water. I mean, I hate it for 30 seconds, but then I don't mind it. And it might be good for me. So I
Well,
do
yeah.
it.
But that's
But I don't call it a vagus nerve stimulator.
right. And a cold shower is different than going into one of these plunges, putting your whole body in there. Wow. I could not do that. Plus, as a cardiologist, I got patients who, if they did that, they could have an MI. They could go into vasospasm and get in big trouble. So, and a lot of people don't even know they have arterial disease. So, okay. Now, another big thing these days about the vagus nerve that people with sensors are measuring, wearables, whether it's a ring or whether it's a mattress sensor or a smartwatch, is heart rate variability, and they're equating that to everything. In fact, ChatGPT Health made that like the number one thing about a person's health is how is HRV, and there's an article from the Washington Post about that this week. Anyway, what is your sense about this measurement that has very difficult literature associated with it, and obviously has a lot to do with the autonomic nervous system?
Well, here we go again, right? um, let's start with what's really easy to understand, and I think probably pretty important. So we can't measure the vagus nerve directly in humans. We don't have the technology. We can put electrodes in animals and we can measure individual fibers and groups of fibers, and we can get a lot of interesting data about how the vagus nerve works by recording its activity in animals. But we don't have the tools to do that yet in people. They'll come someday, and there's some experimental tools on the West Coast that are being developed in laboratories. So it'll happen. But until then, the absolute best indicator of a person or a mammal with increased vagus nerve activity is that their heart rate is slower. And that's because one of the branches of the vagus nerve, it goes to the heart, and the fibers traveling in that branch tend to slow heart rate. So that's interesting and pretty easy to understand. I mean, it doesn't mean if you gargle or hum that you're stimulating specifically those branches to your heart any more than if you gargle or hum that you're specifically stimulating the branches to your pancreas or your spleen either. But at least we know there are branches that go to the heart and slow heart rate. So that's interesting. The second thing I find incredibly interesting is back to your world. The Framingham study showed that the single predictor of longevity in a population was the population with the slowest heart rate lived longer than the population with the fastest heart rate, regardless of other conditions. And that study and that exact finding, with a few minor tweaks, was replicated in the French study, which was what, 4 times bigger or something, tens of thousands of patients. If you had a slower heart rate, if the population with the slower heart rate lived longer than the population with the faster heart rate, all cause mortality. So that's interesting. So is it possible that those people with increased vagus activity are protected against inflammation and have less heart disease? Maybe. It's an interesting hypothesis. It's very hard to study, but it's very interesting.
Right.
So what if you can't measure it, and heart rate is confounded by all kinds of things like activity and stress and other things. So what else well, you can measure heart rate variability. Now, heart rate variability, the problem with measuring heart rate variability comes down to a very simple understanding of what it is. It's a statistical analysis using things like Fourier transformations of individual bits of data, which are the amounts of time between individual heartbeats. So you're looking at the summation of the individual heartbeats as a statistical phenomenon. And the reasoning is that when the vagus nerve fires, it prolongs the time to the next heartbeat. So you get a longer duration between heartbeats. And because the vagus nerve fires intermittently, then the more vagus nerve activity you have, then the more variability you will have in the instantaneous heartbeats. That's the fundamental principle of the relationship of the vagus nerve to heart rate variability. And in opposition to this, you have the sympathetic input to the heart, accelerating heartbeat, which is also going to be dependent on activity and your state of mind and stress and whatever else is going on. So what you do with this information, and here's where it gets up to now,
Yeah.
it's easy.
Yeah.
Up to now, it's easy.
Right.
What you do with this information is subjected to algorithms. You have an algorithm on your watch, you have an algorithm on your wearable, you have an algorithm on your arm, in your bed, you said. We don't know what all these algorithms are. They're all proprietary.
Right.
So now you go into the billions of web impressions on vagus nerve and heart rate variability, and you come up with a lot of what I find to be very confusing information that you alluded to. And here it comes down to what I was pleading for a half hour ago. We just really need more large, well-controlled, well-randomized, statistically meaningful clinical trials to answer specific questions about the relationship of heart rate variability to health. I don't think we have a good understanding of the underlying mechanisms.
Yeah, and I think this is why it's so crazy that the outputs from various, as you say, proprietary algorithm companies are giving people information that, your heart rate variability is bad. Your stress is up. It may not, I know a lot of people using the rings, and they say they're constantly in stress because it's for heart rate variability. Well, I don't know if that's really true, but that's, of course, that kind of output they get. It's using a measurement with a lot of uncertainty, giving people, health advice. I think we'd like to have more vagal tone. Maybe that's why you take cold showers, but have a good way of measuring it. And I would just submit that HRV, you don't want to hang your hat on that because there's a lot of uncertainties, right?
I think that's very well said, and I think that at the end of the day, you have devices with different algorithms that are all proprietary. So if you have money and you want to spend it on wearables, and you think it makes you feel better to know your heart rate variability. Great, I'm not. It's fine. That's what makes the world go round. People do different things, but I think if you're planning your life's medical course around it, then because you think people understand it, then I think that's not right. Then it should be let the buyer beware.
One of the things that I like to look at, and I like interested in your opinion, when you do exercise, you get your heart rate, really jacked up. You're doing a really good, and how fast do you recover? To me, that's a more useful because if it takes you a long time to get back to your basal heart rate, that's not a very good sign of your autonomic nervous system functionality. It's qualitative, and you'll see maybe some outputs about that. But that to me is a pretty good indicator of fitness and functionality of the autonomic nervous system. I don't know if you think that's true.
I do think it's true, and I don't think there's an exercise physiologist on the planet who would disagree with you with the importance of the recovery to baseline, or the importance of a carefully and well measured VO2 max as an indicator of health. And I think there are various exercise advocates recommending different strategies to get more VO2 max, or to improve your VO2 max, or to improve your recovery. I'm not expert on that, and I
Yeah,
don't know enough about.
that's
But
another. because the VO2 max that you get out of a smartwatch is very inaccurate, and it's from walking. That's not the same as going to a physiologic lab and getting a tight fit mask and getting the real deal. And I don't even know if you want to go bother with that. But yeah, that's another thing that is being used a lot. Not that that's connected so much with the vagal nerve per se, but these are misleading miscues that a lot of people are getting regarding their autonomic nervous system function.
Absolutely. I mean, and I did specifically say a laboratory-based, well, carefully controlled VO2 max test. I don't know how my watch thinks it's getting its VO2 my VO2 max when I walk the dog. I
No,
don't get that.
yeah,
No.
I have patients that are coming to me and say, "My VO2 max is dropping. I'm getting really worried." And
But
you they're
but
watching.
let's but Eric, can we talk about the Far Side cartoon with the sheep exercising on the treadmill in New Zealand? Because I think it's relevant to this.
Sure.
So so another, I think, huge misnomer or misrepresentation by some, that when you exercise, you turn on fight or flight, true, and you turn off parasympathetic, false. You don't turn off rest and digest when you turn on fight or flight. You don't turn off fight or flight when you turn on rest and digest. This is everywhere on social media, and there's no evidence for it. Zero. I mean, it's a totally subjective thing how you feel. I feel rested and digested. Great. I feel fight or flight. Great. Okay. But that doesn't have anything to do with what is happening in your nerves. And the proof of this came just a couple of years ago, from a study in New Zealand where the researchers put sheep on an elevated treadmill and had them run, which I just think is a Far Side cartoon. I think that is a riot. If Gary Larson hasn't done one and he's listening, he should draw that, right? So, what the question was, when the sheep are exercising, what's going on with their autonomic nervous system? And what the results showed is that, as you'd expect, when they're jogging along, the sympathetic fibers, the adrenergic fibers to the heart, increase their activity, heart rate goes up, blood pressure goes up. Fine, check the box. When they looked at the vagus nerve and measured the activity, it also went up. And when they block that activity, either blocking the nerve with drugs or cutting the nerve, what happened was the cardiovascular performance of the sheep declined. Cardiac output went down and coronary artery perfusion went down. That means, which is what you would expect if you were designing the system, that the fight or flight and the rest and digest are acting in synergy to optimize cardiac output and cardiac function, which is exactly how you would design it if you were the great designer. That's how
Yeah,
you would design it.
yeah,
And that's how insulin and glucagon work. That's how clotting and fibrinolysis work. Everything, the opposing things work together to get a fine balance. And so that's actually a really important point and leads to where you started all this, is with POTS. In COVID, what we saw, convincing evidence that it started with autopsy studies actually of people who died from COVID in Spain, that their vagus nerves were damaged in a number of those people. And so it raises all kinds of important questions where you began this. If you have a damaged vagus nerve from COVID or some other post-viral syndrome, or from, probably not trauma, but if you have this kind of problem and the vagus nerve can't work, well, there's no evidence that exercise is going to restore it. There's no exercise that a cold shower is going to enhance it or fix it. And so it really does come back to a plea for more research and a plea for more understanding in some who are treating POTS patients, that we don't understand that condition. And it's not a simple on and off necessarily of autonomic, whatever that is, on and off between autonomic balance. But we really should be studying these neural mechanisms underlying POTS, because some of those POTS patients may have damage to their vagus nerve, and some of those POTS patients may have damage to their sympathetic nerves. We don't know. We
yeah.
don't know.
Which could explain ultimately. I hope that the study will be done with the device implant, where some people will respond, hopefully, and POTS can be terribly refractory, difficult to treat, and others might not because of that point you just raised. Now, just in wrapping up, one of my colleagues here got the Nobel Prize for the piezo 2 receptors. And, that, of course, is Arnab Patapushan. Uh, that connects with the vagal nerve. The vagus nerve uses the piece. I mean, the piezo 2 receptors are everywhere, but that is, of course, you might want to just comment about bringing those two things together.
Well, it's been brilliant work that he did, and has been celebrated by neuroscientists and now also by immunologists and physiologists around the world for the reasons that you implied. The nerves are sensitive to vibrations through these receptors, and we and others are now using focused ultrasound to which is a form of ultrasound very similar, slightly different, but very similar to what is used to visualize a fetus in the womb, or gallstones, or kidney stones. And this focused ultrasound can be used to go through the skin and through the tissues. And in contrast to a TENS unit, which stimulates and sends electricity across the skin and in all different directions and places, the focused ultrasound can be focused, as its name says, and you can focus it on the vagus nerve in the liver or on the vagus nerve controlling the splenic nerve in the spleen. And as I said, we and others are looking at this as a way to vibrate or activate these nerves with non-invasive devices. These are still early days, but my colleague who runs the lab with me at the Feinstein Institute, Sangeeta Sivan, and our other colleague, Stavros Sanos, they just a couple years ago published a paper in Brain Stimulation showing that you can use focused ultrasound to activate the inflammatory reflex in human volunteers. And when they did this, they suppressed inhibited cytokine storm in these volunteers using a painless, non-invasive ultrasound handheld probe. You could imagine a day in time. They've also done it. Sangeeta and Thomas Huerta and others in my lab have also done these experiments in obese animals and shown that by activating the vagus nerve with focused ultrasound, you can normalize insulin resistance and blood glucose. You can decrease food intake, and you can accelerate body weight loss in obese cafeteria-fed mice and rats. And so the question is, can we learn from these kinds of approaches to target the vagus nerve in metabolic conditions that may or may not be similar to or the same as targeting the vagus nerve for immunological conditions? There's tremendous. I mean, you said it in the very beginning. What's so exciting about this, Eric, is that I'm not I'm not pushing snake oil here or selling anything. Hypotheses that
Yeah.
can be subjected to experiments and to clinical trials. And the challenge we have now is and sometimes this is the hardest challenge in science, isn't doing the experiments anymore. And we've got all the technology, certainly. The hardest challenge is asking the right question. What would be the clinical trials we could do now that would have the biggest impact? And what would be the mechanistic experiments we could do in the lab now that would get us faster into more clinical trials? That's what has me excited. That's what keeps me up at night.
Well, I mean, I think, just to pull it together, you really deserve tremendous credit, Kevin, for bringing in the field of the inflammatory reflex, using a device that could be implanted that could knock down inflammation, as we've seen for autoimmune diseases. First one out of the block, proven, rheumatoid arthritis, many more to come, likely inflammatory bowel disease and many others. But what you've also, I think, confronted, it's taken you decades to do this because we live in a world of pharmaceuticals, right? And pharmaceutical companies, the device companies that are trying to do this stuff are, like the Rodney Dangerfields. They don't get a lot of funding. They have struggled long, many years. It's a big slog. But I have to say, if I had an autoimmune disease, if I had to take a drug that would knock down my immune system and put me in some risk versus having an hour outpatient implant, I might take the implant, right? So the problem is, we don't have enough interest in this bioelectronic treatment, and it's partly because the device industry, overall has a tougher mission. And you, in a way, have proven that look, you had this hypothesis nailed 20, 30 years ago, and it's only taken until 2025 to finally have an FDA indication and a first, tier, first rate randomized sham controlled trial. So I thought I'd just get you to comment on what's it like to try to advance a hypothesis in the world of devices, because it just seems to be a much bigger um, obstacle.
It I have to say there were plenty of times when I coulda quit, in that
Yeah,
30 years, as you say, but the reason I don't quit is the same reason you haven't. I mean, we meet our patients, they need something, and every patient I've ever asked about this option has said, "If it works, I want it."
yeah,
And we have data now. We have 30,000 people who tried to get into the trial for 242 spots. So patients want this. You know, I've been very lucky. You give me a lot of credit. Thank you. I have 1,000 scientific co-authors, and if you wrote and it likes to start companies and make clinical progress on the business side, if you wrote papers with them, I'd have another 1,000. That's what. And so you talk 30 years, but it's 2,000 people that you work with over
yeah,
over 30 years in order to get this thing to happen. And so I'm sitting here talking to you, and all along the way, I've enjoyed this. I mean, I love doing science. I love waking up in the middle of the night thinking maybe that experiment today will help this patient tomorrow. And I'm the luckiest guy to be able to do what I get to do. The patients are the ones that are the heroes. They don't choose to have the disease, and they sign up for clinical trials. I mean, they sign up and say, "Yes, doctor, you can implant this thing in my neck, and we don't know if it will work." They're the heroes. They didn't. They
yeah.
they didn't choose the path that they were pushed on. So yeah, it's been a long ride. It's been a long ride. There is the there is now the opportunity, I think, under Set Point Medical's leadership with this new immunoregulator to turn the tables on what you said on the pharma industry dominance. I mean, I'm sorry, Eric, if you and I were inventing anti-TNF tomorrow, oh, we go to the FDA and say, we got a drug, it's costs $100,000 a year. It's going to have black box warning. Some people are going to die from it. Oh, and it's invasive. It only works 40% of the time. What do you think the FDA would say to us with those drugs today? They would say, get out.
Yeah, yeah. Well,
So
it was
there's an opportunity now for these devices to rewrite the book.
yeah, up until the GLP-1, Ozempic, Humira and the TNF blockers were the biggest drugs in the world for sales. It
Yep.
shows you how big a deal autoimmune diseases are, the burden in our population. So anyway, I just want to say, the book is phenomenal, but what's even more impressive, Kevin, is that you persevered and you tested the hypotheses and you led to breaking out of a new field. And I just think it's in the early days. I think there'll be a lot more autoimmune diseases. We're already seeing a randomized trial in refractory depression and other areas. So anyway, congrats. It's fun to discuss this with you. I learned a lot, and I know everyone else who joined today did as well. We'll post this with all the links that we've been talking about, and hopefully we'll have a chance to revisit all this progress in the times ahead. So thanks very much, Kevin.
Eric, thank you for having me on. Thank you for all you do to make the world more informed about science and medicine. It makes it a better place. We're grateful to you.
Well, thank you. Keep up the great stuff. Bye bye now. Thanks everyone for joining.